standing until they're seated.
Please be seated, ladies and gentlemen. Professor Deborah Meyer, Executive Dean, Faculty of Science. Professor Annah Moteetee, inductee. Prof. Ben van Wyk, our respondent and also a rated researcher from the University of Johannesburg. Senior leaders of the university, members of Senate and other academics, distinguished guests, ladies and gentlemen, Sanibonani, goeienaand, good evening, Tobela. It's indeed a great honor and special privilege for me to welcome you to the Professorial Inauguration lecture of Professor Annah Moteetee. As I do so, I wish to express a warm welcome to her loved ones. I'm just going to ask them to raise their hands that we can also recognize them here. Warm word of welcome to you all. Special guests and her colleagues. This is indeed a proud and joyful, yet a landmark moment for all of us, for you, Professor Moteetee, and of course for us here at UJ and higher education in South Africa and beyond. The inauguration of professors is a public ceremony in which newly appointed professors are inducted into office and deliver the inaugural address. The ceremony has its roots in the medieval university and serves multiple purposes. Firstly, it's an expression of welcome and an entry for new professors joining the circle of colleagues who are already professors. And secondly, it provides a platform for the professors to display their expertise in the discipline and showcase their research. Today we gather to bear witness to the entry of our colleague to the illustrious community of scholars at the university. It is a celebration of the contribution to the discipline and ultimately the impact on society. Professors provide the university with its identity, character, and academic legitimacy and integrity. The inaugural lecture is a rite of passage following the confirmation of the appointment of the person as a professor. This evening, as we will listen to Professor Moteetee as the gown goes to town. And what we mean by that, ladies and gentlemen, it's that the power of the inaugural address is when the expertise that is showcased here tonight reverberates beyond the corridors of the university within society. It stands out as a moment of pride for the incumbent, the family, fellow scholars, the university, and ultimately society. For the German philosopher and diplomat Humboldt, a university referred to as the world community of scholars and students engaged in a common search for truth. Newman talked about teaching universal knowledge. Recently, universities has been viewed as instrumentalist, serving the purpose of the economy or utilitarian in purpose. I would hope that we can break down and break out of these narrow conceptualizations and reflect on the university as contributing to the public good. Edwards said in an article titled On Defiance and Taking Positions, and he offers a formulation of the ideal role of the true intellectual, and I quote as follows: one who commands a vast knowledge of her discipline, one who is rigorous in the analysis of literature, who views being an intellectual as a vocation, the intellectual who considers it's necessary to step into the public sphere and to speak truth to power, namely to question, interpret, and understand authority rather than consolidating it, to step out of the boundaries of the academy to connect oneself, to affiliate oneself, to align oneself with an ongoing process or contest of some sort, perhaps with the aim of improving the lot of the oppressed. The intellectual who functions as a kind of public memory to recall what is forgotten or ignored, to make the connection that are otherwise hidden, and to provide alternatives for mistaken policies. End of quote. It remains then for us as a university with a Pan-African vision to drive our mandate as intellectuals and as professors. So we do raise the following question: how do we embrace the role of a professor as a disruptor while continuing to be a flagship carrier of our related disciplines. This evening, we will listen to Professor Moteetee as one further step in the journey of being a professor. This is a journey which does not culminate once this lecture has been given, Professor. It is a self-reflective pause to the journey of the professor, with a promise of more to enrich our minds and simultaneously contributing to the rich intellectual body of work in the discipline. Let me now invite the Executive Dean, Professor Deborah Meyer, to introduce our inductee. I thank you. Ria Laboga, Siyabonga, baie dankie.
She graduated with a biology chemistry major at the National University of Lesotho in October 1988. In 1989, she was appointed as a teaching assistant in the Department of Biology in the same institution. In 1990, she was awarded a British Council scholarship to further her studies in the Department of Biological and Chemical Sciences at the Queen Mary and Westfield College, University of London, under the guidance of Professor Geoffrey Duckett. She obtained an MPhil degree in botany in October 1992, and upon her return, she was promoted to the position of lecturer, where she lectured various undergraduate botany courses at all undergraduate levels. During this time, she also served as head of department and was elected as a member of the University Council. She also represented the National University of Lesotho in several government advisory committees and served as the alternate country coordinator for the Southern African Botanical Diversity Network. In January 1999, she registered for a PhD degree in which she conducted a taxonomic revision of the legume genus Melolobium in the Department of Botany at one of the University of Johannesburg's legacy institutions, the Rand Afrikaans University, under the supervision of Professor Ben-Erik van Wyk. In January 2005, she joined the faculty in the same department of the newly formed University of Johannesburg as a technical assistant and herbarium curator. She taught the foundation in bioscience at the East Rand and Soweto campuses and was promoted to the rank of senior lecturer in 2006. In 2011, she was awarded an NRF Tutuka grant for 3 years, which enabled her to kickstart her project on the taxonomic studies in the tribe Phaseolae of the plant family Fabaceae. She was promoted to associate professor in 2013 and to full professor in October 2018. Professor Moteetee has supervised and co-supervised 2 PhD, 7 MSc, and 10 honors degree students to completion, and she is currently supervising 6 PhD, 9 MSc, and 4 honors students. She has published 40 peer-reviewed journal articles and presented 38 podium and poster presentations at national and international conferences, including the biggest botanical conference in the world, the International Botanical Congress. Her main research interests are the taxonomy and systematics of African legumes and other understudied genera, as well as the ethnobotany of Southern African plants, with an emphasis on the ethnomedicine and food plants of the Basotho people. From 2008 to 2011, she served as a member of the South African Biodiversity Information Facility Steering Committee and represented the country at the 15th Global Diversity Information Facility Governing Board meeting held in Arusha, Tanzania. She served as Vice Dean Teaching and Learning for the Faculty of Science from 2011 to 2015, and also as Acting Executive Dean of the Faculty from August 2013 to September 2014. She is currently the head of the Department of Botany and Plant Biotechnology. She enjoys cooking, listening to music, and reading Paulo Coelho when time permits. She is married, has 4 children, including her late sister's son and 5-year-old twin grandchildren whom she loves dearly. I think we can give her a round of applause. And I think you'll agree with me that we look forward to hearing Professor Moteetee profess. Thank you.
Acting Vice-Chancellor, Professor Swart. Please allow me to observe protocol. Good evening, ladies and gentlemen. Thank you very much for coming. Let me take you through the journey of my topic, from plant diversity and traditional knowledge to chemicals and microbes. Let us see what all of this means. Right, so I'm sure that most people will ask, why botany? A lot of people don't even know what is botany in the first place. So why study botany? So perhaps it's what the doctor ordered. As you can see there, I'd like you to see a botanist. Okay. So maybe that is what the doctor ordered. Right. So when preparing for this lecture, I wondered where to start. Then I decided I should start in the middle, because if I start at the beginning, then we'll be here until the cows come home. So I decided to start in the middle. So that is where it started. So armed with my BSc degree in biology and chemistry, I was recruited to join the National University of Lesotho as a teaching assistant on condition that I actually take up botany and plant taxonomy. So I said, well, okay, let's give it a try. And here I am today. I think all of us have aspirations. So here I've put some of my aspirations as a young girl growing up. So as you can see there, I'm sure you can see most people aspire to become a medical doctor, so that's what I wanted to become. But I am a botanist. Without revealing too much my age, when I was growing up, there were only a few things that you could do and that your parents could be proud of you. A lawyer, a doctor, an engineer, probably a pilot or something, but there wasn't a lot. So I wanted to become a doctor, but I am now a botanist. So at the bottom is reality. After I joined the National University of Lesotho, I was awarded a British Council scholarship, and I wanted to join the most well-known university in botany. That was the University of Reading. So that was my aspiration. I applied. I got admission, but somewhere in the middle, my scholarship was moved to the Queen Mary and Westfield College of the University of London. So that's where I ended up doing my master's degree, or an MPhil. Thirdly, I love orchids. So as a botanist, I always wanted to study orchids. Perhaps because they're the most beautiful flower, they have the most beautiful flowers. Also, the family in which the orchids belong, the Orchidaceae, is the second largest flowering plant family with 26,000 species. But just the beauty of it, I wanted to study. But I ended up with Fabaceae, and I have not regretted it at all. So I enjoy working with the legumes. So you can see it's not too far from the Orchidaceae. It's the second largest plant family, but most importantly, whereas the orchids are only mainly just useful as ornamentals, the family Fabaceae actually is the second most important economically family. It includes the beans, the peas, the soybeans. So after the grass family, this is the second most important family in the world. Right, so after I came back from University of London and I went back to the National University of Lesotho, I decided at some point I need to expand my knowledge. So I decided to look for a supervisor. And one of my colleagues said, I know exactly where you must go. You must go to the Rand Afrikaans University. There is a brilliant man there who is a plant taxonomist, Professor Ben Eric van Wyk. And so I met Professor van Wyk. He was happy to supervise me. I'm sure you can see the resemblance here. So he introduced me to this genus, legume genus, which is called Melaleubium. Even my children know Melaleubium because it soon became my friend. Because I spent at least 4 years studying this plant. So Professor Van Wyk and this plant actually shaped my career, which ended up into two focus areas: plant taxonomy and ethnobotany. So I just want to delve a little bit. Please, if you'll indulge me, I'll just to try and answer the question, what is taxonomy? So taxonomy has a few things that go with it. Firstly, you need to find the plant, right? So to find the plant, you need to go to the field. So you find nice places to find the plants. And this is some of my field trips with my students. And in the process, you also might find your most favorite animal in the world. For me, it is a giraffe, which is why it is there. And as some of my students discovered, if you don't find your plant will find you. This is the reason for this particular slide. The owner of these legs will soon be revealed. So sometimes you look for a plant, you look and look, and you think it's not there. You get into the grass, and you come out like that, and you're like, wow, there's my plant. The biggest challenge with trying to find plants, especially in South Africa, is the fact that most of the land is actually privately owned. So sometimes you drive for hours and hours. You get to the place, it's a fence, and it's locked. And you're like, okay, what must I do now? But that's the first thing of taxonomy. Secondly, you need to identify your plant. And the plants have got a number of cues that one can use to identify. One can use the vegetative parts. For example, you can look at the way the plant grows, the growth habits. You can look at the leaf structure, the type and distribution of the hairs. So for example, here, some of the plants will tell you, here I am, and I am a blackjack. Right? So that's very easy to identify, some of them. And as we work on legumes, you can see the diversity is so interesting. Sometimes the plants grow flat on the ground. Sometimes the plant twine around others. They climb around others. Then you can look at the leaf structure as well. You see there's all interesting types of morphologies. And I must upfront acknowledge Professor Van Wyk for his photos. I think one of the requirements for being a botanist is that you must be a very good photographer, but I think I'm still a good taxonomist, but I'm not a good photographer. Then once you've collected your plants, maybe you bring them to the lab, you can also look at, for example, the structures on the leaf surface and so on, the hairs, you'll do your microscopy, and all of these characters you start dissecting, you will actually be able to identify the plants. You can also look at the reproductive structures of the plants. For example, you look at the flowers. Can you see how beautiful the Fabaceae, the legume family flowers look like? So that is why I have not regretted studying this plant. So you can look at the shape and location of the flowers. How are they arranged? Are they arranged in a stalk like that? Are they individually arranged? You can look at the characters on the fruits. So you can really see that these plants are actually legumes, because of the pots there. So the legume family is quite big. Some of them are edible, the majority of them are not edible. So most of these are actually wild legumes. Then you can even open the pots or the legumes and look at the seeds as well. So the seeds can also help you identify the plants. You can also do some microscopy. Sometimes there's subtle differences that you can have a look. You dissect the flowers and you see some interesting characters. Then you can use what are known as identification keys. So for example, if you've got plants like this that look exactly the same, how do you then begin to identify them? So you can use this identification keys that will take you to the different species according to their different characters, as can be seen here. Classification. Classification can be at different levels. Here I've put the simplest and the quickest way to be able to identify plants, for example. So obviously to any person who knows the sunflower, you can see any plant that looks like that belongs to the sunflower family. Any plant that has flowers like that belongs to the Fabaceae family or the legume family. And then, of course, different genera, different species, and so on and so on. So there are different levels of taxonomic hierarchy. Here I've just represented the family level. Then you name your plant, and here is my close friend, Melaleubium microphyllum. Microphyllum is a species, which means that it's a Melaleubium with very small leaves. Microphyllum means that. And then you have to give the right species name. And taxonomists sometimes make it difficult for other taxonomists. You can see that this plant here has got so many names, then you have to decipher as a taxonomist which is the correct one. So out of just 10 names, sometimes 20 names, you come up with one and the correct one. So there's only one correct plant name. And then you describe the plant, of course. How does it look like? The growth form. This one is a spiny shrub. The leaves, etc., etc., the flowers, and so on and so on. So that is taxonomy. So the question is, why do we need taxonomists? If you can look at a plant and tell that it belongs to the Fabaceae or it belongs to the Asteraceae. So our country, South Africa, is very unique in terms of biological diversity. It is the third most biologically diverse country, only after Brazil and Indonesia. It comprises 100,000 species, and here we're talking about all living organisms. So if you consider our species, Homo sapiens, if you consider that we are one species, we are totally outnumbered, because there are other 999,000 or whatever, 99,000 other species apart from Homo sapiens. So South Africa's biodiversity actually contributes 10% of the world's plant species. So you can see that we have such diversity in terms of especially plants. There is also high levels of endemism. At least of the plant species, 65% of those only occur in South Africa. Now that is quite something to be proud of. It is the only country in the world to hold or to have what is called a floral kingdom. That is the Cape Floristic region. Now, how do we know all of this? Without sounding arrogant, this is the work of the taxonomists. Only taxonomists can tell us actually how many species do you have in the country. Now, I'd like to introduce a very important organization. It is called the South African National Biodiversity Institute. They produce, so they are custodians actually of biodiversity information. They must make sure that there is access to the information and record it, make sure there's proper documentation of the biodiversity. So as custodians of biodiversity, they publish a number of things. So they share the information via a number of ways. One of those is by publication of what are called floras or checklists, for example, of the country, sometimes of the sub-Saharan country, and so on and so on. Now, this is the work of taxonomists. So taxonomists will actually contribute here. So if you take a pick and open this book, you'll find my little contribution of the genus Melaleubium. So you can see that we do contribute. Also, the taxonomists, this work is also important for conservation purposes. So if you don't know how much diversity you have, how would you know what to conserve? So those are some of the importances. So I'd like to talk again about Sanva, because our work really contributes a lot to their work. So they published a number of things. In this particular publication, it's a research strategy that they publish. In this, they mention their mandate. The mandate of Sanva is actually the coordination and promotion of taxonomy in South Africa. That is why Sanva has to work hand in hand with plant taxonomists. There's also, they say that there is a need to buy in by taxonomists into the objectives of the Sanva. In this particular 2015 strategy, they mentioned that they want to produce an online flora that can be accessible to everybody for the country by 2020. And I'm very proud that actually Dr. Marianne Leroux, who's in the room, is actually very responsible for making sure that the South African plant biodiversity is available online. And I'll talk about that a little bit later. So we do contribute. And this other article from Sanva, again, they state the dire need for plant taxonomists in the country. So we are actually a scarce species. There are only 35 professional plant taxonomists in the country. So if you consider there's 100,000 species of plants, 21,000 species of plants, you can see that there is really a dire need. And they go on to say, even if the human capacity to conduct taxonomic research in South Africa was doubled, there would still be work for taxonomists to do so. Whether or not they will employ the taxonomists is another matter. But there is a need for them to be. So here I just want to introduce a little bit of my work, and just to show how Melaleubium actually helped with my journey on plant taxonomy. So this was a first taxonomic paper from my PhD, and this paper, together with that one, published in the South African Journal of Botany, actually won a Compton Prize for the best articles published in the journal in 2006. It's a very long time ago, but that actually gave me courage to continue working on plant taxonomy. So the CV did mention that I work also on the tribe Phaseolae. The tribe Phaseolae is where all the beans are. Whether wild, whether cultivated, all the beans belong to the tribe Phaseolae. So I have done a lot of taxonomic revisionary work on some of the genera, so I have revised. So here I just mentioned a few. I like to mention Sphenostylis because it's a nice little group to work with. So this was a paper that we published with Professor Van Beek. So you will notice that the scientific or the generic name actually are derived. Most of them are actually Greek. So Sphenostylis is actually sphenos is wedge for this style that is shaped like a wedge. So there it describes some character of the plant. That is what is nice about the Greek names, although they are very difficult. So in that paper we provided a distribution map, we provided distinguishing characters. Although there's only 2 species, we provided also a diagnostic key which tells us how the 2 species differ from each other. Another example is the genus Carnevaleax. I enjoyed revising these species because many species actually have agronomic, they have economic, they have ethnobotanical, they have medicinal, they also have nutritional value. And this group was very nice to revise because, as I said, plants do have cues. For example, the sword bean, you can see that one can really distinguish the different species mainly according to the color of the seeds. One would have expected, for example, that species with white flowers will probably have white seeds, but it's not like that. So I found this group very, very interesting. And you can also see it's very widespread, but some species, for example, this first one, the sword bean, actually is called Carnevaleax africana because it actually is restricted to Africa. So you can see on this distribution map. But you can see the jack bean is very widely distributed, and so is the sword bean. Some of these beans are actually eaten in other parts of the world. So I think it's probably about time that we start developing some of these plants, especially because they do have a nutritional value. Right. Then, of course, you don't work in isolation, so I just want to mention briefly my collaborations. The biggest taxonomic work that I'm doing is actually on the genus that is called Rincosia. This is the snout bean. It is a large genus comprising 240 species. It's widely distributed, but its center of diversity is actually Brazil and Southern Africa. So together with collaborators and students, we have described a number of new species. So obviously this adds to our diversity as well, to our plant diversity as well. So these 2 papers were describing species that were found in the Western Cape mainly. And then with Dr. Leroux, who's in the audience, we also looked at this group. So when a genus is too large, it's divided into sections, sometimes into groups, sometimes into complexes. So the one very well-known botanist who worked mainly in tropical Africa, worked on this genus as well, but in tropical Africa. And he said, when he looked at this group from tropical Africa, he said this is one of the 3 groups of maximum difficulty in terms of trying to decipher which species is which, what is the species, and so on and so on. And then you can see there were 19 names that we could find in the literature. So, but actually there is only one species. All of these refer to one species. So we managed to actually come down to one species, and we recorded one new species, we recorded one new variety in this particular paper, and this is how the plants look like. Still continuing, so of course another aspect is student training. So I want to just talk about some projects that we have with students that we have. Some of them are ongoing, some of them are completed. So Rincosia, as I mentioned, is a large genus. It's divided into 4 sections. So the one student actually revised these 4 sections. So you can see there a plant taxonomist in training. And this actually study resulted in 4 papers, 2 with this particular student. And these plants mainly are climbers. They twine everywhere. So sometimes you can actually miss the plant if you go look for it in the field. And then we also look at how the species relate to each other. Where do they sit in the phylogeny? So we did some molecular work with Ms. Manielo here for her master's. So in South Africa, as I mentioned, the genus is divided into 4 sections. So she used 3 gene regions, 2 chloroplast regions, 1 nuclear region. And the chloroplast regions actually told us absolutely nothing. Whereas with the nuclear region, one could actually try and make some sense of what is going on. And we did this, thankfully, in Professor van der Bank's lab. And we also looked at another group, the Rincosia minima, as the name implies. These plants have got very, very small flowers. And again, when looking at this group, Verdcourt said this is unquestionably the most difficult group within the genus from the point of view of taxonomic complexity. So it is so complex because the species and the plants are very, very variable. If you go to one region, you find different leaf arrangement. If you go to another region, you find different, but actually it is the same species. Common theme is actually the tiny little flowers. But this student here, Mr. Tukhwane, discovered that distribution is actually very, very important. So he discovered 1 new species and 2 new varieties. And these are the new varieties that he discovered. One occurring isolated in Namibia, so that is one variety, but he also noticed that the most important character was actually the presence of the clans, where the clans occurred, what type of clans they are, but most importantly, the distribution. This is the one variety, this is the other variety. So it is not just distribution, it is not just characteristics that you look at, you can also look at the distribution, and then it can tell you something. Then once you start looking closely at the plants, you can actually see that they are actually different, and there is no overlap at all in terms of characters and in terms of distribution as well. We have a PhD student. Now he's revising the whole genus. He's doing molecular work. He's also doing morphological work to see where Rincosia fits. There are 2 clear groups in the type section, Rincosia. There is the twiners, the ones that twine, that climb, and so on, and there are those that grow erect or sub-erect. So far, 4 new species have been discovered in this group. One of those has been published in this particular paper. We I mentioned that this particular genus is centered mainly in Southern Africa and in Brazil. So we got a shock of our life when we attended the International Legume Conference in Japan recently, and we found that actually the Brazilians are working on our plants. And we said, please hold on, these are our plants, and I'm glad we attended that conference, because then we were able to establish collaborations that they said that we're happy to work with you. So we are now pushing on with those collaborations. So I'm hoping that represents some form of GES, Prof. Then we're also looking at, so the South African National Biodiversity Institution has also produced a priority list for the genera that need to be revised, that need to be studied. So bit by bit, slowly, we're looking at those plants. So one of those is Trifolium, but the main reason why Sanva approached us to look at and study this particular group is because it's not very well represented in Southern Africa or in South Africa. There are only 5 indigenous species, and 15 of them are exotic. So because of the fact that many of them are exotic, there's a fear that they might have an invasive potential. So we're looking at that to see, do they have a potential to be invasive? But alas, although the work is still ongoing, the research is ongoing, we've actually found that you cannot find this indigenous species everywhere. The exotic species. What you find are actually the native species. This particular species, you will find it everywhere. We drove miles and miles and miles of kilometers in KZN looking for these species, and everywhere you go, every locality, you find that particular species. So although natural or indigenous vegetation cannot be invasive, this particular species, I think, can be declared as invasive. But of course, research is ongoing. So we actually revising the genus. We also want to evaluate the infrageneric diversity. We're going to look at the populations to see how diverse they are genetically. So the work is ongoing. Mr. Mercado here is doing that work. So finally, the owner of those legs that we saw is here. So Kubo is revising Cynoclosum. Cynoclosum is also listed by Sanva as a priority genus that needs to be studied. So Cynoclosum is actually hound's tongue, so called because it's got the leaves that look like a dog's tongue, and the texture as well. So cynos is actually refers to dog, and closum refers to the texture, the hairy texture. So cynoclosum, we call it buhume in Sesotho. Buhume means something that sticks to you. So because of this reason. So as I said, this particular group, the fruits are the ones that stick to you. So the fruits are the most important diagnostic character for this particular group. And when Kumo did some scanning electron microscopy, she saw that the fruits were very, very useful. And the hairs that are found here on the fruits are very, very distinct. So you can see, for example, that this particular arrangement represents one species, this particular arrangement represents another species, and so on and so on. So when you start looking closely, these plants look so similar that for all we know, they could be one species. But once you start looking closely, and you need a scanning electron microscope to be able to tell the difference between the different species. And here are just examples of those fruits. So plants, of course, use fruits as a form of dispersal mode. But of course, for us as taxonomists, they're very, very important. The structure is very, very important. That's what we can use. Then we're also looking at the Lightening Bush. This is also listed by Sanva as a priority genus in need of taxonomic revision. It's relatively a large genus. As you can see, it is an African genus. So Africans must study this group of plants. It is actually mostly found in the Cape region as well as Western Asia. We're looking at especially the KZN complex. There, the species are quite complicated. We found this particular variety, for example, Clutia abyssinica variety, Usambarica, occurs in KZN, but it also occurs in East Africa. But when you look closely at the species, you find that they actually look different. And the varieties find that they look different. So this means that this is probably a different species or a different variety. So studies are ongoing so that we can see what is going on there. They differ in the leaf shape, for example, the size of the leaf. So they must probably be something different. So we're looking into that as well. Right. So now with all of that in mind, usually being visionary as we are, we then at some point came up with global excellence in stature. And we need to publish in high impact journals. We need to have high citations. You need to have a high H index. So UJ wants to be up there with the rest. And I think we've embraced that and we want to do that. So now, to be a taxonomist or not to be a taxonomist, this is what has led me to this question. So I will just now in the next slides talk about my work in ethnobotany. So we published a paper that we didn't think was very, very important in a very low impact factor journal, local journal. And look at that. This is my paper that won the Compton. This is a very prestigious award in terms of botany. It has 8 citations, the last time I checked, published in 2006. And this one, the medical ethnobotany of Lesotho, we just did a review. And my supervisor used to say that we need to publish bread and butter papers. You have to have good papers and bread and butter. So this we published as a bread and butter paper. And look, the last time I checked, published when? In 2011, it already has 46 citations. This another ethnobotany paper, published in 2015, already has 36 citations. So they both have overtaken the taxonomy paper completely. So now one has to decide. Here is Sanva requesting us to train taxonomists to study these plants. And here is UJ saying that we need quality. So one needs to decide. So, but now I just want to talk about my work a little bit and tell you what I've decided to do. So what is ethnobotany? So there's different definitions of ethnobotany. One author, Hirschberger, in 1896, you can see that ethnobotany is actually a very old area of study. Say that it is the study of plants used by the primitive and aboriginal people. Right, so that is ethnobotany. Robbins in 1916 said that it is the study and evaluation of the knowledge of all phases of plant life amongst primitive societies. You can see it's actually linked to primitive people, aboriginal people, primitive people, and so on and so on. Ethnobotany is a term used to refer to the academic discipline that deals with people's interaction with plants. So maybe I think that is probably a more acceptable definition. But what I like is this last definition. So he defines it and then he says, in other words, it is a marriage between cultural anthropology, botany, study that investigates the roles of plants as medicine, nourishment, natural resources, or gateways to the gods. I think that about sums what is ethnobotany. Right, so I am hoping that somebody will actually coach me. So this is my quote that I think I should come up with. So I have read a lot, obviously, about ethnobotany. So reading about plant use in the literature was actually an eye-opener. just to see how important it is. But recollecting my grandmother, my mom, and all the elderly people in the village actually using the plants is actually priceless. So I'm hoping somebody will cope. So where does the chemistry and the microbe come in? If you're a botanist, what do you have to do with microbes and chemistry? This is the definition that actually shows the link between all of those. Ethnobotany, by its name, is an interdisciplinary science that combines anthropology and botany. But to carry it out well, it needs to involve collaborations between many other disciplines, including chemistry and pharmacology. So that is exactly what it says about chemicals and microbes and the link between those two. Right, so how is it done? So there are two main ways in which you can do it. You can conduct ethnobotanical surveys. You go out to the community to find out how these plants actually are used. So where we had one project, Mr. Papo here, who comes from that area, Hamashashani, went back to Hamashashani in Limpopo to study the plants, to talk to the people. So he conducted surveys by way of questionnaires, and he recorded 45 species. He recorded 12 new uses, which were not recorded elsewhere in literature, and actually recorded one new species record in South Africa's Materia medica. And this is this particular plant here, not that little guy, but that particular plant here. Nobody else had recorded the use of this plant. And so this is where the microbes come in and the chemicals as well. So we tested with my collaborator, Professor Van Vieren there from Wits University. We tested 43 of these plants for their antimicrobial properties. 7 of those showed very good activity against the pathogens that we tested it against. 21 of those showed moderate activity. Of surprise to us was this plant that you know very well. It showed very low activity against respiratory tract pathogens, but it is actually used mainly for that. But I want to think it is actually the essential oils that are used that are important, especially in medicine. We also did some qualitative phytochemical screening, and we discovered an array of chemicals. So the thing is, these chemicals, alkaloids, flavonoids, terpenoids, saponins, all of them have actually been found to have medicinal properties. That is why the plants are actually used medicinally, and that is why probably they are effective. And also, I think we must not be alarmed if there is no antimicrobial activity. It simply probably means that the active compounds may be present in insufficient quantities, because many a times when we test these plants, some of them don't show any activity at all. So what must we do? Must we go out and tell the traditional healers that your plants don't work, so use something else? But there are reasons why that these plants don't work. So these are some of the plants that he recorded. Some of them you might have seen, and so on. And then another ethnobotanical survey we took we happened in Maseru in Lesotho by that doctor, now Dr. Kosi, and he recorded 80 plant species used in that particular area. And in that survey, he recorded 8 new plant records, 146 new uses which were not recorded anywhere else in the literature. So that is quite staggering amounts of new data that we discovered. What is particularly interesting is the fact that these plants are mixed with animal products, such as, for example, milk, animal dung, chicken gizzard, rock rabbit urine, etc., etc. And I think that Professor Van Vieren, I think when we test some of these plants, we must actually add animal dung, chicken gizzard, and so on when we test them, because maybe that is how they work. And these are some of the plants that she recorded: the old wood, buckle plant, spotted color, common African hyacinth, and the wild mint, and so on. So we put those plants through scientific validation. 44 of them were tested for antimicrobial properties. So we tested them against respiratory tract infections, pathogens, STI pathogens, skin pathogens, and so on and so on. 7 of those showed very good activity against the microbes. 15 showed moderate activity. We also tested these plants for their toxicity, just to see how toxic are they, how safe are they. And of the ones that we tested so far the STI plants. 20 of those actually were found, only 3 of those were found to exhibit some cytotoxicity. The rest of them are safe to use. One plant, interestingly, when we tested one plant, we found that the leaves were actually much more toxic than the roots. But interestingly, traditionally, they use the roots. So most probably they already know that actually the leaves are much more toxic. So let's rather use the roots. So actually, traditionally, it's common practice to use roots, although it's very bad in terms of conservation, because naturally the roots are actually much more they are chemically rich. So that is why they use. So the traditional healers and people that use these things probably they have some foresight, they have some insight actually. And they refuse if you tell them you must use leaves rather instead of roots. The other reason why they use roots is because roots are hidden away from all evil spirits and so on and so on. So I grew up in a very rural village where each and every plant had some use or other. And I read about this in the literature, and that, of course, corroborates the fact that these plants are, in fact, used traditionally. So if one look at, because ethnobotany is quite wide, it could talk about medicinal plants, food plants, and so on, functional plants, but my main focus has been ethnomedicine and the food plants. So I mentioned to you that actually Professor van Beek and Melaleubium brought me to plant taxonomy and ethnobotany as well. So when we studied Melaleubium, we found that one species is called Musa Balu in Sesotho. And Prof. van Beek recollected somebody telling him about Musa Balu. And then he asked me, what is this Musa Balu? And I remembered very well growing up, if there was a bereavement in the family, people would be running around to look for Musa Balu. So it was used to calm down people that was actually under duress as a result of stress. And when we looked, we found that actually 15 species belonging to the legume family are actually used as Musa Balu. They are all called Musa Balu. Musa Balu means one who turns around the heart, literally. That's what it means, right? So in this particular paper, we discuss the possible mode of action for those plants, right? So they either act as sedatives to relieve anxiety and stress, for example. In the cancer bush, which is that one, the cancer bush has been tested and it has been found to have this particular compound, the amino acid, which is actually an antidepressant. It's been tested and found to be like that. Then they act as adaptogens. They help increase the body's resistance to stress, or they act as immune stimulants, or they act as digestive bitters, which of course act on cardiovascular system to decrease the heart rate. Of course, if your heart rate is decreased, then you are you're relaxed a little bit. So Melaleubium is the one that actually brought us to ethnobotany. And these are some of the species that are all known as Musa Balu in Sesotho. Another very interesting plant is Cercia irosa, at least to me, because when I was growing up, this plant, my grandmother used to send me to the bush to collect this plant. This very uninteresting plant. And she used to mix it with her snuff. So she would grind her tobacco and she used to mix. So I was very interested to find out what is in this plant that actually my grandmother will send me every single day to go and collect in the field, and she mixed it. So it is also used medicinally as well. It is used to treat gastrointestinal problems. It is used for respiratory tract infections. It is also used actually for tanning animal hide. In the olden days, it was used like that. So we did some anatomical work. We did some, we distilled the leaves to find out if it has essential oils, because if you crush the leaves, they've got this smell. That's, so we wanted to test it, and we did a lot of dissections. We tested which chemicals it has, where they are located, and so on and so on. So the histochemistry showed that there were quite a number of compounds. For example, essential oils, alkaloids. These might be actually responsible for their therapeutic uses. It had high amounts of tannins. Of course, that is why it is used for tanning. So you can see that actually the primitive people were actually quite clever. They could see the plant has tannins, so we can use it for tanning. We found some promising antimicrobial activity against the pathogens that we tested it against, and thus it validates the traditional use for gastrointestinal. Another very interesting plant is Pantanesia prunelloides. It has several names that shows that this plant is very, very well known. It is very well known in Southern Africa, and you can see all of the names they point to fire, right? Regardless of where they originate. So you have Sitima Mollo, that fire extinguisher, that's literally what it means. Iqima Mlilo, that's exactly what it means. And somebody who speaks Afrikaans can say that one for me. So also fire, right? So this plant, the main use, it's used for several medicinal purposes, but the main use is actually for the treatment of heartburn. Because if you have heartburn, you feel like you actually burning. Yeah. So people simply chew it. So we need to know. So the work on heartburn is very interesting for me, and I'm working with Professor Kinfe here from chemistry to see if the plants actually have antacid properties. So we're doing a lot of tests. The plant is not only used for heartburn. You can see it's used for many other reasons. The other reason for its name could be because it relieves the burning pain of boils. But I suspect maybe it is because of its use for heartburn. So we're going to use this artificial stomach model, which Prof. Kinfe is building for us. We're going to test the properties of these plants. This work is interesting because nobody in South Africa is actually doing this work. So for us, it will be new. So we had better move fast with that lady there. And these are some of the plants. And believe it or not, blackjack is one of those that are used for heartburn. One would have never guessed, and it's never been tested before. So these are some of the plants that we're testing. So you must just watch the space. And another plant is called wild scabious, Scabiosa columbaria, known as Silumi in Sesotho, because the main use of this plant is actually for treatment of period pains, which are known as Silumi in Sesotho. So a lot of these plants are actually named for the function that they perform. But it is also another very well-known Southern African plant. You can see the uses there. It's got endless uses, but one of those is actually heartburn, so that is my main interest. It's used mainly for female reproductive problems and so on and so on. So we're testing the plant also for its antacid and acid neutralization properties. But the other interest with this plant is that it's used also in combination with other plants. So together with Prof. Sandy, we're going to determine if these plants have got any synergistic effects if they are used in combination with each other for the treatment of various problems, especially infectious problems. And these are some of the plants with which it is mixed with. Another very interesting plant from anecdotal experience is this one, Sishoku, lion spoor, milkpole. This plant, when I was growing up, I promise you it works. If you had cracked heels, really you take the milky latex and you use it, it would work. So I am very interested on working with plants that are used for wound healing. So we have another project with Buhle there. She started for her PhD looking at plants that are used for wound healing purposes. But the main reason for that, the main inspiration for me was actually to test this plant, to see if it has wound healing properties, and I'm hoping it should have. And these are some of the plants that are used for that particular. So she's done now a literature survey, and she discovered 345 plant species are used for various wounds. 40 of those are unexplored scientifically, so those are the ones that we're exploring to find their antimicrobial activities, their anti-inflammatory properties, because as you know, wound healing is quite a complicated process. And actually the actual wound healing properties themselves. So this is what we're doing. So from medicinal plants to soap plants, my particular interest was this plant, which is called Sesepa sadinoa. Those who speak Sesotho will know Sesepa means soap. So it's called Sesepa sadinoa because it is used traditionally as a soap substitute. So I'm also interested to find, do these plants have saponins? Because saponins are the chemicals that actually have or make the soap foam. So these plants, the soap property of plants is reported to be due to the presence of the chemicals called saponins, so called because they've got soap-like properties, so they have got foaming properties. So we want to test these plants. We're testing these plants, and we've seen some very promising results. Some of these plants have got very high saponin amounts. We're also screening them to see if they do have, because you use soap to cleanse with the hope of removing germs. So we want to test, are they active against skin pathogens as well. So as I say, there have been some promising results in this regard. Right. So when I grew up, we also ate a lot of plants. So 2 projects that have just been completed, wild indigenous fruits. So when I was growing up, we used to take those fruits as green as they are, and you dig a hole, you put them there. So this was pre-harvest. You collect them, you dig a hole, you put them in there, cover them with soil. 2 weeks later, you come, the fruits are ripe. So my main interest in this particular project was to see, do wild fruits have any nutritional value? Do they give us any nutrition? So that particular plant was the one that actually influenced me to do this. But a lot of other fruits obviously are eaten, and we tested quite a number of those. We tested the proximate analysis, sitting in the audience with Dr. Kayitesi from Food Tech and Biotech. So we did some of this work, and we found some very, very interesting results. For example, my particular plant had the highest phosphorus content of the ones that we tested. This is higher than the plum. Can you believe it? This plant, the fruit, had very high protein content, higher than the guava. It also had the highest carbohydrates, at least of the ones that we tested. This particular fruit, the wild date plum, had the highest fiber, higher than the apple. The low felt milkberry had the highest vitamin A content, higher than beef liver. So I would suggest, instead of eating beef liver, we should just eat that one. But those who know this fruit is very difficult to eat because it's very tough, actually, and there's not much there to eat. Maybe that is why some of these plants are not commercialized, even though they are very, very nutrient rich. The sour fig had the highest calcium, but unfortunately, all of the fruits that we tested had low vitamin C content. But obviously, one can eat a lot of oranges to try and make up for that shortage. Then we looked at food plants. We looked at wild indigenous vegetables, because every time I came back from the fields with my grandmother, we would collect these vegetables and eat them, of course. So I wanted to know, what do they give us any nutritional value? So we tested the plants. We did the same as we did with the fruits. So citrulus lanatus actually had very high protein content. The nightshade, the black nightshade, had the highest crude fat content amongst the one that we tested. And then Sonchus tragianus, which is not here, had the highest moisture content. And then, of course, we still want to compare with cultivated vegetables, because a lot of previous studies have actually shown that many of these wild vegetables are actually much more nutritious than your cabbage and your spinach and so on and so on. Right, so in the concluding remarks, I ask myself, so where to, obviously? So what do you want to do next? So obviously, despite the lack of citations from the taxonomy papers, it's my national duty to actually keep my expertise alive, my area of interest, so to train taxonomists. A lot of the people that I've trained actually work for Sanva, so that is good. So I think one should continue doing that. So continue to revise the genera that I need of taxonomic revisions and so on and so on. The genus Alchemilla and the Rosaceae, Miletia, and then these are some of the people that have started. Now, this is very important, I think, for IR, I think, because so our work contributes to Dr. Marianne Roux's work. So they want to, one of the strategies of Sanva is actually to place 22,000 species, the information, all the information, the species descriptions, diagnostic keys, distribution data, and so on, online. Now, all of that work is actually going to contribute to the world online flora of all known plants. There are going to be 400,000 plant species. So I think in terms of 4IR, we are contributing probably in a small way to this particular project. Now, in terms of ethnobotany, documentation obviously is very, very important, because as we all know, traditional knowledge is passed from one generation to the other orally. And this traditional knowledge is fast disappearing with the passing and the death of elderly people who have this information. So there is a need to document these plants. So speaking Sesotho and coming from Lesotho, the Free State obviously is the natural choice, because it's very, very understudied. Not a lot of work has happened there. So I want to continue with the documentation there. Numerous medicinal plants were found to be antimicrobial active. So the question is, so what? What do you do with this information? Professor Van Beek hosts an annual conference, Indigenous Plant Use Forum, where we all meet and discuss this. And one very famous South African professor there always asks, we come to this conference every year and you tell us how important these plants are. You tell us how microbially active they are. And then what? There are so few South African or African plants that are actually in the market out there, either as medicinal plants or soap plants or whatever the case might be. So you have all of these promising data. Then what? We must do something. That is what he's been telling us. We must do something. Can we get these plants out there, at least? But those who in the know will know that the competition is very high. It's very, very tight to try and compete with the European market, American market, and so on. But we have to start somewhere. We have all these promising results on screening of saponin content and so on and so on. So, for example, when we wanted to collect plants for soap, to screen them for their soap properties, this is the response we got from the Department of Environmental Affairs in KZN. They said the project has bioprospecting implications, so you must not start here, you must start with the minister. Because as soon as your project has some implication of bioprospecting, you must get a permit from the minister. So you can see how difficult it is. The other question that we always get asked when we present at conferences is that, so do you now want to take all the plants and turn them into pills, right? You want to take the plants and put them in a bottle. But that is, a lot of people miss the point. That is not the point why we're studying ethnobotany, right? The plants, the information is disappearing very fast. We need to document it. The second reason is that people don't use, a lot of people don't necessarily use plants because they can't afford Western medicine. They use medicinal plants or traditional healing because it is supposed to be holistic, right? It does not just treat the physical ailment, it treats the whole body and the spirit and so on and so on and so on. But we must do something with plants. So according to DIA, research for purposes other than bioprospecting is defined as a specific activity, not a bioprospecting project. So we had presented our application as a specific activity research, not for bioprospecting. So that tells us that maybe we should be doing bioprospecting. Especially my main interest is this last 2 points, antacid effects and also the possibility of soil plants. So hence my question here. Bioprospecting is a long and tedious process, Professor Van Beek will tell you. These are some of the things that you need to do. So you need to apply for a bioprospecting permit from the minister, not from the department. And those are some of the things that bioprospecting entails. But there is a plant that I showed, it's called blue squill. So I am predicting in the future there might be maybe blue squill soap. Are there possibilities of antacids from some of these plants? So these are some of the questions that I asked. Some fruits and vegetables prove to have good nutritional value, some better than their commercial counterparts. So what next? So we need to test, this is what Dr. Kaitesi told me, the food biotechnologist, we need to test these plants then for their dietary qualities. Can the people digest them? But I think we more or less already know that because we have been eating these things. But scientifically, we need to validate that actually are the nutrients that we found, are they available to the body once you've ingested these plants? Does the body have the right enzymes to digest these plants? And so what are the possibilities? Maybe jam, you could put some of these fruits into bottles and so on, but definitely not that one. So, with that, I would like to make some acknowledgements. I would like to thank God Almighty for always responding to my emails and my WhatsApp messages. And I thank my parents for putting me in this world, and my lovely husband, my best half, my children, some of my siblings. The children have actually helped me, supported me. Some of them forgotten at school while I was studying and so on and so on. So thank you very much for forgetting that you have been forgotten at school. I would like to thank all colleagues, former Executive Dean of the Faculty of Science, the current Executive Dean of the Faculty, Professor Meyer. Thank you so much for your support, really. Thank you. And my colleagues, Prof. Van Beek, Professor Whitehead, Professor Van der Bank, Prof. Tilney, for all you have contributed somehow to my research, at least. So I thank you so much for that. And my other colleagues, thank you for being there. And as head of department, I wouldn't have been able to continue with my research if I didn't have such a good department. These people jump, even if I don't say jump, they jump very high, so they do deliver. So thank you very much, colleagues sitting there. And also all heads of departments, and particularly those that are here for all your support, for smiling, and we all face the same problems, and I think we have some kind of an unknown club that we discuss our problems. And all my collaborators, Professor Van Vieren sitting there at the back, Prof. Boatwright at UWC, Prof. Leroux at Sanva. And I'd like to thank faculty office. Some members have really been helpful. Deans from other faculties who I have worked with when I was actually vice dean and when I was acting as dean, and two of them who are here. Thank you very much, Prof. Betty, Prof. Sachi, thank you very much for your support and thank you for being here. And NRF for funding, of course, you need the money. And then I need to thank, actually, Prof. Swart was also a dean, and we were put in the same team, actually, when we had that training with Gibbs, and I think we worked very well. But today you're representing the university, so I wish to thank the University of Johannesburg for allowing me to continue with the taxonomy work, although it does not have any citations at all. But I wish to thank you very much, and I just want to mention to Prof. Swart that we have also started collaboration with Ghana, University of Ghana. So I'm thinking that also is contributing towards the Pan-Africanism initiative that UJ has. And I thank you very much, ladies and gentlemen. Left the last one for last. Of course, the stars of the show, it would not have been possible. These are some of the postgraduate students, some new members as well, some old members, and so on.
Good evening, everyone. I have the honor and privilege to be the respondent to Professor Moteetee's beautiful lecture. The first thing I'd like to say, this impact business, let's say citations, you know, there was once, long before the young people in the audience time, there was a famous rugby player. And he used to, what we call, play for the pavilion. Eh? So he would have his comb in his sock. And then he was playing fullback. He would kick the ball, and then he would take out his comb and comb his hair and put it back. So he was playing for the audience. So that's one strategy in science, is to play for the audience, play for the university. But there's another strategy that was very nicely expressed, beautifully expressed by the person who received the honorary doctorate from the university recently, Professor Feringa. And I can contest to this. The reason why we do science and why we do research is for the excitement of discovery. That you get goosebumples when you come and you see, here is a plant that no one's seen before. It's obviously new. Or you see a process that's completely new. Science is about novelty, about new things, new discoveries. So that is, of course, the main motivation. Also why we do taxonomy, is to discover new species and to describe them. So Anna, I agree with you totally, your synthesis that you've made. We have taxonomy, that's absolutely necessary, because we live in a country that's the third most biodiverse country in the world. Someone has to make an inventory of the plants we have. So that's a very, very basic, it's the most basic part of botany, is what plant is that? What plant is this? Someone's got to know. The second part is, what can we use it for? That's the ethnobotany part. So those two aspects are exactly what you presented to us. And that is correct. What plants do we have? We don't want to lose them. We want to protect them. We need to know, but you have to give them a name so we know we can talk about them. And then what can we use them for? So this extreme biological diversity that we have, botanical diversity, and the extreme cultural diversity makes for a very, very complicated system, because each culture uses plants in different ways and different plants. And that is unexplored territory. I'm telling you, it's shocking to see how little has been recorded. Traditional knowledge, indigenous knowledge, is rapidly fading. But perhaps the most important aspect of all, the most important impact of all, is Annah Moteetee's contribution to a new era in ethnobotany, where it is not clever people from Europe studying primitive people, but it is where it is primitive people studying primitive people. That is a major mindset change, and I think that is the future. And in that way, Annah will have impact far beyond citation indices. So I thank you very much, Annah. Congratulations for becoming a professor at the university. I'm very proud of you.
Ladies and gentlemen, I think you will agree with me that our professor can profess. I think you agree with me. And while they make their way forward for us to rope our colleague, why don't we give her another round of applause? So, ladies and gentlemen, you might have noticed that Professor Moteetee came in without a gown. Part of this process is that following an inaugural lecture, that we're going to rope her now as recognition for this wonderful contribution that she's made here tonight. Let's stand here and you can all get into the photo. Join us, please. There we go. So please allow me a few concluding remarks. I know that it's been quite a long day for many of you, but it's been such an invigorating lecture. Let me start off by thanking Prof. Moteetee for allowing us to accompany her on a journey to this night here tonight, where she started off as a young girl, collecting various issues for her mother, other family members, but ending up here tonight, showing to all of us the fine scholar, great academic, and it's indeed a privilege for us to welcome you to the cohort of professors at the University of Johannesburg. Thank you very much. It's wonderful having you here with us. Professor Meyer van Wyk, for your response, remarks. It's really much appreciated, but also alluding to the novelty and the significant contribution of the work that is being done, and how to also contribute to the great ambitions of this university, to international university of choice, but also understandings that our footprints and that our roots are anchored in Africa, and this contributes to that as well. Thank you very much for that. But please also allow me, and I think the colleague has alluded to, from the university side, to thank the family. Your contribution, your support. For those of us in academia, we understand it's long hours, it's hard work, it's lots of commitment, and we know there's sacrifices on the side of the family. So thank you very much for your support over the years. Let's put our hands together for them as well, ladies and gentlemen. And then to Professor Meyer, the Dean of the Faculty of Science, Executive Dean of the Faculty of Science, for creating an environment where such talent can flourish and come to the fore. We really do appreciate your contributions in creating that enabling environment. Now, I please ask that you rise as the procession leaves the venue. Thank you very much, ladies and gentlemen.